just run through some of the things that we've done in here, right? So the first thing we talked about was movement. And a big part of what we talked about the movement was the basic ganglia, all right? So you guys should be able to draw the basic ganglia like this, normal basic ganglia. And so we have the cortex sending input in to the caudate and putamen. So we have our 4 boxes here. We have the subthalamus below, and we have the thalamus, which is our output back to the cortex. Right? The input and the output is excitatory. Right? All the connections within the basal ganglia are inhibitory except the subthalamus. So the subthalamus and the thalamus both have excitatory outputs. All these areas are regulated inhibitory. We have 2 regulating dopamine pathways coming in to the caudate and putamen. Into the caudate, it's inhibitory because it's D2, but into the putamen, it's D1, which is an excitatory dopamine. And when everything's flowing and working well, right, this helps us regulate our motor movements, to get smooth fluid motor movements, and it also helps us cognitively regulate our cognitive functions. Then, we talked about 2 different disorders that affect the basic ganglia. What were they?
Parkinson's.
Parkinson's and Huntington's disease. What happens in Parkinson's disease?
Doesn't it take away the caudate?
What's that?
Does it take away the caudate?
No, that's Huntington's disease. So, take away dopamine, that's Parkinson's disease. So Parkinson's disease, we lose the inhibition and the excitation, and so you should be able to map what happens next, right? If we lose inhibition, this gets more active. Its output's inhibitory. That means we're going to shut down the external lobus pallidus, right? So it can't inhibit these areas. So there's a lot more excitatory input into here. Over here, we took away the excitation, so there's less activity. So we're not able to inhibit this very much. So the internal lobus pallidus gets heavily stimulated. Its output's inhibitory. That's going to shut down the thalamus and not allow us to have a proper signal back to the cortex. That results in the movement patterns that we see for the inappropriate movement patterns for Parkinson's. In Huntington's, we lose the caudate. Dopamine's fine, but we lose the caudate. And when we lose the caudate, we cannot inhibit this anymore. So it gets really active. It's out of control, right? And it shuts down the subthalamus and the internal lobus pallidus. And so now we get not much regulation, no inhibition of the thalamus, and we get too much output, and we get the excessive movement patterns that we see in Huntington's disease. Good? All right. This is just another graph that we drew and talked about where we have this range of sort of normal dopamine coming in. When we have Parkinson's disease and we start to lose those neurons, we fall below. And at some point we start to have symptoms of lack of movement. And so the treatment, L-Dopa, is going to boost, when we take that medication, it's going to boost our dopamine levels. We want to keep the dopamine sort of in between this normal range. But sometimes it's hard to control that, and initially, once they take the pill, they may actually have some excess movement. So they're up here and there's too much dopamine. And then as the drug starts to wear off, they go back through that period. So we really want to try and keep them cycling within this period and not falling down here and not being treated. Cool? All right. Now this is one. That all right. I'm just going to put the basic ganglia there on this brain, right? With obsessive compulsive disorder, I talked about that on our post-lecture video, right? But with obsessive compulsive disorder, we have obsessive thoughts. We have obsessive thoughts in a loop, right? From the orbital frontal cortex through the basal ganglia back to the orbital frontal cortex. And normally when someone has an anxious thought, the basal ganglia, part of its regulation, helps shut down that thought and helps you regulate that. And what we know in obsessive compulsive disorder is that there's a little bit too much influence of dopamine here, and the basal ganglia has a hard time stopping this loop. And so they have an obsessive thought, did I lock my car? Comes back around and says, oh my God, did I lock my car? And he keeps doing it round and round. And even like when they go back and lock it, that simple act of locking it can't break this loop. It keeps going round and round and round. So what happens is they end up doing some kind of motor back, right, from the motor cortex, where they lock the car 10 times, right? And that happens to correlate, doing this repetitive behavior over and over again, with stopping the loop. The loop stops then. And it's not really that this stopped it, it just happened at the same time. And so there's a reward, right? So now I'm no longer anxious. And so this, locking a car 10 times, gets reinforced as the thing that stopped the obsessive thought. Right? Yes?
Does caffeine make some people anxious because it increases dopamine?
No, just because it's a stimulant, and so it just increases activity in the brain, kind of hyper. And so you can have something like this become sort of hypersensitive. Yeah. Just because it's a stimulant, not because it doesn't actually act on dopamine. And we'll talk about caffeine when we talk about sleep wake cycle in the next section. All right, so this is sort of what's going on, we think, in an obsessive compulsive disordered brain, right? So we get now the compulsive act that becomes associated with stopping the obsessive act and gets reinforced. So the next time that there's that loop gets going, the person starts trying to lock their car 10 times, thinking that that's what's going to stop the thing, right? Ways to treat this. There's 3 ways to treat this. What are the 3 ways to treat this? Cognitive behavioral therapy is going to break the association, the reward, and teach how to stop the loop cognitively. Right? Good. What else?
SSRIs,
Drug-
serotonin.
Yeah, drug therapy. And SSRIs have been used probably more commonly we're giving dopamine antagonists. But we're trying to act here to shut down the circuit pharmacologically, right, with drugs. Right? And if drug therapy doesn't work, and cognitive behavioral therapy doesn't work, and this is out of control and the person can't function in life, what's the last option?
Is it the exposure response therapy?
No, that's part of the CBT, but good. Exposure response prevention therapy is part of CBT. No, not ECT. Surgically break the loop, right? So we come in and lesion the pathway so that the loop can't be active, right? And so that's what is shown on this slide, right? So I wanted to sort of draw that out and explain it, because just looking at this, it's hard to walk through it, right? But we're good? on obsessive compulsive disorder? And how the three different ways each act on a different, the three treatments each act in a different sort of place in a different way to stop bullying. All right. Sensory systems. Any questions on sensory systems? We have physical stimuli. Transduction. How does it get turned from a physical stimulus into a neural signal? Coding. How is labeled line or a cross fiber pattern, or both, represented in each sensory system? Then we have a pathway. How does it get from the receptor into the primary cortex? And then we have areas of the brain and their functions that we talked about: primary, secondary, association cortices. Yes?
Can you go a little bit more detail in neural code?
Yeah. So we have two different types of neural coding: labeled line and a cross fiber pattern. Labeled line means that any signal in that neuron is labeled for a stimulus. So we could have a neuron that is labeled line for movement, acceleration or deceleration in a particular direction, right? Because we have three different semicircular canals. We also can have cross fiber patterns. Cross fiber patterns, the brain has to look at the pattern of incoming signals in order to understand what a stimulus is. So while we do have labeled line for the three directions of the semicircular canals, whether that signal is excitatory or inhibitory from baseline is going to indicate if it's acceleration or deceleration. So that would be a cross fiber pattern for acceleration and deceleration. We talked about in the taste system, we have labeled line signals for salt and sweet, where if there's activity in those neurons and it's a sweet neuron, we know that sweet is on the tongue or salt is on the tongue. Different neurons that are labeled, they only respond, those neurons only respond to salt and sweet. But we also have a cross fiber pattern neurons that respond in a different pattern to help us discriminate between salt, sour, and bitter. Does that help?
Thank you.
So there's examples here of, for each of the sensory systems of that. Some abbreviations: ORN means olfactory receptor neuron. That's the receptor, olfactory receptor neuron. Taste receptor cell, here TCR, taste receptor cell. So for olfaction, we have 7 transmembrane receptors, right? So those olfactory neurons have receptors on that odorants, the shape of the odorant can activate and cause G protein release of the signal. So we have labeled line signals for each receptor type, right? Each ORN, each olfactory receptor neuron type. But a smell is going to activate multiple of these, and it's the pattern of which ones of these get activated that represents the quality of that odor in our brain. So that's cross fiber pattern. Yes, Emily.
So is it different neurons for cross fiber pattern, or is it just whenever there's combinations of labeled lines?
It's combinations of labeled lines that create the cross fiber pattern. Any questions on sensory systems? Anything that doesn't make sense? So as I'm going through this stuff today, if you're like, this would be a good opportunity for you to be thinking in your head, do I really know that, or is that something I need to review? Do I really know this, or is that something I want to make sure I can go over and diagram and know all the parts to it? Do I really know transduction mechanisms? Right, or is that something I need to review? So be thinking about those things as we kind of cover them too. Anything else on this? All sensory systems go through the thalamus, right? So you should know what part of the thalamus belongs to each sensory system, right? So that information is charted here, right? Excuse me. The VP, ventricular, is both vestibular and somatosensory. Because remember, vestibular kind of doesn't have a primary cortex. It's kind of going to the somatosensory cortex, but also to things like your superior colliculus and cerebellum for balance. LGN for vision, MGN for auditory, VPM for gustatory. I don't think I said this in class, but the thalamus for olfactory is medial dorsal nucleus. I told you this stuff was not easy. It's confusing for everybody. All right, let's look at leptin first, right? So we have our fat cells, and when we have increased leptin, we have decreased leptin. And so, leptin can actually act on both of those systems in an arcuate. So here we're going to have the arcuate. And we're going to have okay. When we have a lot of leptin, right, that's going to act through Tom C. Clark, right? That's going to then decrease feeding. And the problem is I was trying to draw that linearly. But if you look at this, right, there's lots of activity on both those areas, right? So the MPY, if you're flipped around here, so the MPY is on top and the MC is below. But it's not just a linear, like only acting on one thing, and only acting on the PDN or only acting on the ventral median, right? That's why it's so complicated. So let's go back to this slide. Okay. All right, and if we have low leptin, right, that's going to then act to stimulate MPY, right? And we know MPY is going to inhibit the PBN. And when the PBN gets inhibited, that means that we're going to get an increase, right? In eel size, right? PBN. Not the VMH, which is eel number. Yes, increase in eel size. When we have high leptin, right, then through the PBN, right, that this is going to be stimulating the PBN. And when the PBN gets stimulated, this is going to be low activity. Maybe I should have drawn that in red. Right? Low activity, we get that. High activity, decrease in meal size. Good? Better? Clear? Okay, let's look at ghrelin. Let's look at the short-term stuff. If that was wrong for one thing. So when we have increased ghrelin, this is fasting. The longer we go after a meal. This is where we're good. So over there we have ghrelin that's coming in to the NPY neurons to stimulate the NPY neurons. And when we stimulate it, that's a feeding signal, right? Right? So when we have low activity coming out of when we have PYY, this is satiety, we're going to come in and inhibit and that's going to have high inhibition, low inhibition. And so when we have low inhibition, we're going to get high activity, which is going to decrease meal size. Our meal numbers. All right, are we happy now? You feel better?
I think so, yeah.
Delete, throw away all that other stuff. I'll take a picture, I'll put it into the review slides and repost it after class. Okay. Sorry, I did not mean to make that as confusing. This is inherently a little confusing. Okay. So here's where we go. All good on feeding? All right. I didn't put up the other homeostatic systems, but what is it that's important for you to know about each homeostatic system? Set point? What is a set point? That's the variable we're trying to maintain. And so in order to maintain a set point, what do we have to do? You want to keep your body temperature at 98.6, what do you have to know?
Temperature.
The temperature. What is the temperature? We have to have a receptor. We have to be able to detect it. This is our variable target. We have to have receptors that can detect, are we at our target, or do I need to push it down or up? So then we're going to have a regulator This is going to be an area of the hypothalamus. We need to have a regulator that can move it up or down to get back to that set point. And so that regulator then is going to have a response. And those responses are usually both physiological and behavioral. Right? So for body temperature, our set point is 98.6. Where are the receptors? What are the receptors for temperature? We have two. We have a peripheral skin cells that are detecting our peripheral temperature. And then we also have the preoptic area itself can detect core temperature. So we have two receptors, peripheral and preoptic. And so if we're too low or too high, we have two areas of the hypothalamus. The preoptic area, if we're too high, is going to promote heat loss. And the posterior hypothalamus, if we're too low, is going to promote heat conservation. Right? How? That's the regulator. How? What are the responses? What is the physiological response? Yeah, so we can increase our basal metabolism or decrease our basal metabolism through non-sugar thermogenesis, regulating our basal metabolism. And we also regulate our blood flow. We constrict the blood flow or we dilate the blood flow to get it towards the core if we need to conserve heat, or away from the core if we need to lose heat. All right. What about behavioral responses? Change your environment, right? Create heat. Get in a cool environment. Take off your jacket if you're overheating, right? So we have these complex behavioral responses. Same thing for fluid regulation. Thirst. We talked about osmotic and hypovolemic. So we have two different receptors, OVLT for osmotic, subfornical organ for hypovolemic thirst. We have the lateral preoptic area that's responding. And we have a release, we have a physiological response to release vasopressin to either conserve water or stop releasing vasopressin to release water. And we have, we can have actions on salt conservation, because fluid balance really is about salt water concentration. That's our set point. Our set point is, are we hypotonic or hypertonic, or are we isotonic? We want to be isotonic at that 150 millimolar salt level. And if we go low or go high, we have to balance things. And we can balance that by initiating salt appetite and salt conservation if we have too much fluid, or if we don't have enough fluid, we can initiate drinking as a behavioral response, and water conservation. All right. Good, we're good on that. And then we talked about, all of these areas, sort of the complexity of feeding. Other questions? And that's pretty much all my slides. That is, it's not pretty much. Zach.
Say for like the short answer or the free response, like kind of just stated questions. Do you prefer that we put the whole name, like ventromedial hypothalamus, or would BMH be sufficient?
BMH should be sufficient.
Okay.
You can use the initials, that's fine. And the same thing for the nuclei for the sensory systems. If you want that LGN or MGM, that's fine. Yes.
What exactly is the orbitofrontal cortex that I see?
Orbitofrontal cortex does a lot of complex things, right? So it is involved with, in obsessive compulsive disorder, it's involved with those feelings of anxiousness and worry. It is involved with flavor. There's an area of it where all the vision, taste, olfaction, and touch all come together to represent flavor in the OSC. The OSC also has limbic components associated with it. I mean, it does a lot of different things. There's a lot going on. Because remember, you think about, we only have this much of the frontal lobe. All this is motor. So premotor cortex, supplementary motor cortex, primary motor cortex. So we have to pack all that cognition, all that stuff into the tips of the frontal lobes. And so there's a lot going on in those areas. Personality is found up there. Making judgments about consequences is there. Questions, things you want me to cover? Yes, ma'am.
So what does it mean when it's asked for the pathway of the homeostatic systems?
Well, it's sort of like the receptor, the OVLT is going to detect osmotic thirst and then hand that off to, so there's a pathway that's going to go to the lateral preoptic area. And then the lateral preoptic area is going to stimulate release of vasopressin, it's going to initiate thirst. So just sort of the sequencing. That's what's important. Other questions? Got plenty of time. Yes, Alan?
Can you explain proprioceptors again?
Yes. Proprioceptors?
Yeah.
Yeah. So we have two proprioceptors. Muscle spindles, the S is spindles, they detect stretch, the S is stretch muscles. And we have Golgi tendon organs, T and tendon put retention, they detect tension. Right. When does our muscle have tension? When it's contracted or relaxed? contracted, right? So the Golgi tendon organ is a signal to stop using that muscle. The muscle is in use, and then you can stop using it. When is the muscle stretched? When it's not in use, and the antagonistic muscle has pulled it out, right? And so that's going to be a signal saying, hey, you're not using that muscle. If you want to contract it, go ahead and do that.
So it's not a separate thing, it's just this mechanism.
Those are proprioceptors. And they use, right, different neurons. The 1A sensory neuron is for the spindle. The 1B sensory neurons for the tendon organ. And then because we want to be able to report tension at different levels, not just when it's fully stretched and when it's fully contracted, but in between, we actually have these things called gamma motor neurons that are going to kind of tighten up the spindle as it starts to get slack. Sorry, not the spindle, yeah, the spindle. Tighten up the spindle as it starts to get slack. So alpha motor neurons are causing the contractions at the neuromuscular junction, and the gamma motor neurons are contracting the motor spindles. Good? Other questions?
I think there's some stuff that I'm still kind of confused about with auditory sense. Since it's like, I'm kind of confused about the differences in the roles of like the pinna, the middle ear, and the inner ear, because I keep getting those mixed up. So could we go over that?
Absolutely. So there are three parts of the ear with hearing: the outer, middle, and inner. Okay? The outer ear is your pinna that helps collect sound, and the shape of the pinna helps with sound localization going up and down. Vertical sound localization. We talked about sound localization for left to right, and we talked about three different ways that we do that. Interoral time differences, the time that one sound hits my right ear and my left ear. So if it's all the way over here, it's going to hit my right ear first, and then it's going to hit my left ear. And that's done by what part of the brain? It's the first nucleus going up towards the cortex. Remember my little mnemonic device? Uh-huh.
Is it that one?
Yeah.
Like Sonic NG.
Yeah, the SON, that's where neural time differences are happening. Superior elevary nucleus. The SON part, detecting time for left-right sound. Then we also have, for low frequency sounds, which part of the phase, up or down, is the wave in. And for high frequency sounds, intensity. So they're going to get absorbed by the head. So if it's louder in one ear than the other ear, then we know it came from that side. Okay? So three ways we do left-right sound localization. The pinna, vertical sound localization. After the pinna, we go down to the middle ear. The middle ear, job is to take this pretty weak airwave, sound wave, and turn it into a wave in the fluid cochlea, in the fluid-filled cochlea. That's the job of the middle ear. So the middle ear has the tympanic membrane that catches the sound wave, and then the three bones that are going to amplify in a lever-like fashion that smaller sound wave into a bigger fluid wave inside the cochlea. So the middle ear's job, amplify the sound. Get it to the cochlea.
Are we gonna have to know like the names of
No, you don't need to know the names of the bones, just for trivia. And so once the fluid wave is going through the cochlea, it's going to get transduced there. And so the tectoral membrane is going to bounce on top of the basilar membrane at a particular place, depending on what frequency is present. And when it bounces, it causes those channels to open and allows potassium in, and potassium then allows calcium in, and calcium causes action potentials to go to the brain. And if it's, so depending on the frequency, it's going to depend where that tectoral membrane bounces on and activates. And so if it's a 400 hertz sound, it's only going to be here. If it's a 4,000 hertz sound, it's only going to be here. If it's a 10,000 hertz sound, it's going to be way out here. So that's a labeled line. Which neurons are active represents a frequency. If it's below 5,000 hertz, we have enough receptors dedicated to those frequencies that groups of neurons send volleys of action potentials that actually the brain can count in a cross-fiber pattern, and it approximates the frequency 25,000 times a second through that group of neurons. And if one neuron can do that, but a group of neurons can do the volley. Right? So when we have hundreds of neurons, not just 5, but hundreds of them, their activity now looks like the sound. Only under 5,000 hertz. Label line, all frequencies. Does that help, Rachel? Good. Any other questions? Anything else? I'm happy to stay as long as you
Can
want.
you go back to the sensory system slide on a review and talk about the thalamic regions?
This?
Oh, it was the one where it's all 6 laid out? Yes. And this is part where it's a pathway. It's kind of confusing to me.
So, for touch, dorsal root ganglion, dorsal columns of the spinal cord, VP of the thalamus, post-central gyrus, this primary cortex, right? So each of those is going to have a pathway. Here's our auditory, cranial vague, sonic, SON, inferior colliculus, MGN of the thalamus, superior temporal gyrus. Optic nerve comes in, the medial parts of each eye cross over to meet up with contralateral lateral parts. Goes through the LGN and the thalamus to V1, occipital pole. Secondary pathways directly to the superior colliculus for eye tracking or object tracking, and hypothalamus for circadian rhythms, sleep-wake cycle. Olfactory neurons to olfactory bulb, to the MD of the thalamus, to the amygdala and the piriform cortex being the primary. Taste, 3 nerves come into the NST, go to the PDN. There's connections between the PDN and NST and the hypothalamus, so hunger and satiety changes our taste signals. And the amygdala for value, hedonic value of taste. And goes through the VPM, thalamus, insular cortex, and then secondary OFC. Secondary areas are, so primary areas of sensory systems detect the quality of that stimulus, right? The quality feature detection of that stimulus, so particular features of that sensory system. How is the auditory cortex organized? What features is it trying to detect? What are the primary features that the auditory cortex is trying to detect?
Is amplitude?
Amplitude is going to be how much it's stimulated from low to high. But the areas of the cortex are organized according to what? Think about the primary job of the auditory system is to detect what? Frequencies, right? So it's organized from low 20 to 20,000 hertz frequencies. Visual system is really complex. It's detecting lots of features like angles of lines, colors, simple movements, and then reassembling those in its secondary areas. Secondary areas do some kind of processing of those features, right? Some kind of processing of those features. So, Wernicke's area and the angular gyrus in the auditory system are looking for patterns of frequencies that the brain recognizes as language, belonging to language as opposed to some other sound. In the dorsal stream, those secondary areas are looking at how do I respond to this visual object. In the ventral stream, those secondary areas are looking at how do I identify what is that visual object. In taste, OFC is flavor. It's a secondary area. It's processing, it's taking all the taste when you drink lemonade and it's sweet and sour and maybe a little salty, and it's taking all those columns that we're firing for sweet, sour, salt and recognizing that that's lemonade and putting it together with smell and vision and touch. Association cortices are like memories. What is that object? Okay, I know that pattern, I know that flavor, and that flavor is associated with my memory of lemonade. So that's the distinction between sort of primary, secondary, and association. Yes, John?
I'm not sure if you touched this when I was going, but when you go into detail or just like briefly touch on cerebral dominance,
Yeah, so we talked about that when we talked about language being lateralized to the left hemisphere and all other sort of environmental, other non-language sounds being lateralized to the right hemisphere. So the secondary auditory areas are doing different things in the left and the right side of the brain. The right side of the brain, they're recognizing that that's the sound of a motorcycle, not a car. In the left side of the brain, it's doing language. And so that's, we refer to that as cerebral dominance. So the left side is dominant for language, the right side is dominant for environmental sounds. Or we can call it lateralization.
Thank you.
Stay here for 20 more minutes if you have additional questions. Yes, Rachel.
This might be a silly question, but I just have written down volley theory, and I can't remember what that is to save my life.
Yeah. So the volley theory, we just talked about that with the auditory system, right? Less than 5,000 hertz, groups of neurons send volleys of action potentials that mimic that frequency, if it's under 5,000 hertz. So if we have a 2,500 hertz tone present, right, it would activate the 2,500 hertz spot area on the cochlea, which is labeled line. But that area would have enough receptors that they would send action potentials at a rate, that group of neurons, not any one neuron, but that group would send a volley of action potentials to the brain that was 2,500 times a second. And if we change that tone from 2,500 to 2,505 hertz, the labeled line, it's just going to move over just a tiny bit because we only added 5 hertz. So it's harder to discriminate between if it's coming from here or coming from here. But if it's under 5,000 hertz, we can count the difference in those extra 5 volleys per second and be able to discriminate and detect that that's a different tone at 2,505 compared to 2,500. Cool? Good job.
You spoke about this last class, but can you go in more detail about the arm wrestle organ? Essentially, I didn't write it down all the way. Arm wrestle. I know it goes to the pyramid.
Yeah, memorable things.
Okay.
Yeah. So that was a secondary system, right? It's not shown in this slide. And it's mainly in non-human mammals. And so the VNO, femoral nasal organ, is in non-primate animals has a whole separate pathway. And it actually goes to a separate olfactory bulb, and it's a whole separate pathway. And it links right into reproductive behavior. Humans can detect pheromones through their normal olfactory system. We think that it's still acting in a subconscious pathway. You don't smell, you don't perceive the odor consciously. But it doesn't cause overt reproductive behaviors. Now, it can do some things in humans, like help women that are all living together and smell each other's pheromones can begin to have the same menstrual cycle. Right? There's some not definitive research, but some sort of loose research to say that women respond to pheromones from men differently than they respond to pheromones from women, and men respond differently. So there is some changes in like skin conductance, GSR, maybe heart rate, that's subconscious, but it's not like an overt reproductive behavior.
Thank you. And the theory taste receptor cell, can you touch on that a little bit?
What?
The taste receptor cell?
Yep, that's the cell that's going to transduce all the taste. And it's going to happen in different ways for different tastes. So salt and sour are inotropic, and sweet and umami and fat and bitter are metatropic, lock and key.
You said umami, right?
Umami.
Umami, right.
Yes.
Can you talk about the biological correlates to NPH, normal pressure?
Yes.
List?
So in NPH, pressure builds up in all of the ventricle system, not just in one area. So you have the lateral ventricles pushing out from the inside of the brain. You have the third ventricle pushing on the midbrain. You have the fourth ventricle pushing on the cerebellum. You have all the cortex surrounding, all the fluids surrounding the outside of the cortex pushing in on the cortex. And that results in the general dysfunction of the cortex results in dementia, inability to think clearly, and memory problems. The pressure on the basal ganglia in the lateral ventricles results in the shuffling gait and the heart and the difficulty moving. And most importantly, the pressure on the third ventricle results in urinary incontinence, which is what is sort of the symptom that makes it different from Alzheimer's, which has dementia, but not urinary incontinence at the beginning, and Parkinson's, which can have dementia and shuffling, but doesn't have urinary incontinence. And the treatment is simply to relieve the pressure, to put a shunt in, drain the pressure off, and the person's fine. And we think the pressure comes from just that inability to absorb the CSF as fast as it's being produced. So it's slowly just, or that output, the creation of CSF is at a faster rate than the removal of CSF. And so it builds up slowly over time. Is that good? John?
I didn't have this fully written out. I know the C fibers into the dorsal spinal cord in terms of pain signals. What else are the C fibers in terms of?
C fibers are dedicated for pain, right? And they're the unmyelinated signal for pain. And then we have A delta fibers, which are the myelinated fibers for pain. Yes, Rachel?
Can you go over the adipose tissue again, the brown fat versus the white fat? And
So the white fat is what's going to release leptin. Okay? The white adipose cells are our fats, or what we think of as fat storage cells. And they release leptin. Brown adipose tissue are fat cells that have fats, so they have energy in them, an energy source in them, but they're also loaded with mitochondria, which is what makes them appear brown. And they're located near your core organs, and they generate heat and help us maintain our core body temperature.
is that because of the mass amount of mitochondria in there?
Yes, because when the mitochondria become active and use the fat to create energy, that energy creates heat.
Thank you.
Yes. And that's non-shivering thermogenesis. Yes, Frank?
I remember the term free nerve endings. What are those?
Free nerve endings are in the touch system. So we have two classes of receptors. The mechanoreceptors, which are the 4 different mechanoreceptors that have different structures that allow them to detect different touch. And the free nerve endings that don't have anything around them, they're free, right? No receptor structures. And they detect increases and decreases in temperature. So we have a class of warm fibers that detect increasing temperature and cool fibers that detect decreasing temperature. And they also can detect pain. So those A delta and C fibers have free nerve endings as their receptors. Any other questions?
Can you talk about the color there, like a quick second? The color there in terms of the visual, the color there?
The color code,
Yeah, yes, sir.
color coding?
Yeah.
Yeah. So we have 3 cone photopigments. Right, Haley, you talked about this? And they do what colors? Red, green, and blue, right? They're labeled line outputs. But we see other colors that are not just red, green, and blue. So orange or yellow, let's say yellow, is directly between red and green. So if we have equal amounts of medium and long wavelength, we perceive that as yellow light. And that's called trichromatic theory. And from there, from the retina, those outputs go to the thalamus, where they form excitatory inhibitory connections in the LGN, and we get opponent process theory, where you get neurons that respond to blue but get inhibited by yellow, and neurons that respond to yellow but get inhibited by blue, and neurons that respond to green but are inhibited by red, and neurons that respond to red and are inhibited by green. Again, that's a labeled line, but the pattern of those allows us to see colors other than blue, green, and red. Yep.
Can you go over like the regulation? I don't know how to say it, it's like
Poikilothermic?
Yes.
Yes. So what are poikilothermic animals?
Cold-blooded?
Cold-blooded, or they don't have a regulator, right? They don't have a way, they don't have a physiological response to control their temperature internally. So the only way they can regulate their body temperature is through behavior. So they have to specifically move to a different environment in order to regulate their body temperature. It's a good session. Yes, Anna?
I know we've probably like gone through this so many times, but I'm just very, like, I'm still confused on like the vision transduction process. Can
Yeah.
you just go through it?
Sure. So it's a three-step process. The tricky thing is that in the dark, when there's no light present, those receptors are actively inhibiting, they're releasing inhibitory neurotransmitter to prevent action potentials from going to your brain. So if we don't allow action potentials to go to the brain, we're not going to have any visual perception because there's no light. We're in the dark. No light, no activity going on. Now, when there's light, it's a three-step process. So light comes in and activates the photopigment. The photopigment releases a signal that closes the sodium channel. And so the cell is no longer excited and stops releasing the inhibitory neurotransmitter, which can allow a signal to go to the brain because we saw light. So it removes the inhibition. So in the dark, that sodium channel is open and causing the receptor to become depolarized and release its inhibitory neurotransmitter and prevent signals from going to the brain because we're not seeing anything. Does that help? I know it's confusing.
So the sodium channel is what allows or inhibits like
Active in the dark and prevents signals from going to the brain because there's no light.